Hi everyone. Thank you very much for inviting me today to this very interesting event that bridges science and art. It was very, very interesting to hear all the speakers. And I'm Valeria. I'm a PhD student in Sapienza University of Rome, but I work in the labs of the Polyclinic University Gemelli in Rome. And today I will discuss with you all the applications of copper, of the copper element in the biomedical device. And I will also discuss a bit of the atomic properties of this element. I think I will go through my presentation all in English, and then at the end I will do a small recap in Italian. Perfetto. So today I will, as I told you, I will introduce this field, the copper applied for a microbial interface from atomic properties to biomedical applications. So first of all, copper is a quite well-known element, but where can we find it worldwide? The first thing that comes to my mind is that the word copper actually comes from Latin and from the Latin word cuprum, which means coming from the island of Cyprus. In fact, this island was the main source of copper during the Roman Empire. But if we have a look at the history of copper and the usage of copper, it actually goes back in time, and we find the copper as protagonist during the Bronze Age. In fact, in Mesopotamia, which is located in the Asian Asia, in Asia, copper was used as a metal. It was warmed up, and humans knew how to work with it because they discovered that actually copper could be mixed with other metals as tin to create more durable, more strong alloys like bronze. And bronze was used to create all the weapons that could be used both for defending themselves in case of battles or in case of wars, but bronze weapons could be also used to kill animals and so to provide food for the whole group of the small towns. And nowadays, the main producer of copper is in the South America is Chile, followed by another country which is always in South America, which is Peru. If we go through all the atomic properties of copper, we can actually find this metal in the 11th group of the periodic table, and it has an atomic number of 29, and it is reported that the atomic weight is around 63 atomic units of atomic mass. The electron configuration of copper shows that this element has 10 electrons in the 3D and 1 electron in the 4s valence shell. And if we have a more deeper look about how copper appears as a solid, we can see that it can be present as mineral or in powders. The mineral, which is on the top right, has a reddish color, and it has like a coral morphology, while the cuprous oxide and the cupric oxide are two different powders. And you can see that copper can actually change in color. In the case of cuprous oxide, the color is reddish or brownish, while in the case of cupric oxide, it appears to be more blue or even black. And both these powders, cuprous oxide and cupric oxide, can be used as paints for ceramics or glasses, but very interestingly, they can also be used in biomedical applications, as we will see later. Copper is also always associated with blue color, especially when we find it in solution. And we are talking about the X-aqua copper 2 ion, which can react with ammonia and change its color. When ammonia is acting as a base, so in the presence of a small amount of ammonia, the color of the ex-aqua copper ion changes from light blue to a slight darker blue. But in an excess of ammonia, there is a ligand exchange reaction that actually causes the change from a light blue to a very dark blue color. If I ask where do you usually can find copper in daily life, well, the principal answers that you can find is that we can find copper every day when using cents, for example, €1, €2, or €5 cents. But copper is also an element present in architecture. In fact, on the left, I reported a small picture of the landscape in Prague. And if you have a look at the green rooftop, but also if you think about the Statue of Liberty in New York, the green color is caused by the oxidation of the copper layer that is on these architectures. And then if we go more deeper into the biological field, we can see that copper actually has a very important role inside plants, as it can act as a cofactor and it is involved in enzymes activity that regulates metabolisms and cell growth. And copper is also present inside the metabolism of some crustaceans, of some animals like crustaceans, because copper has the role of transport binding oxygen and transporting oxygen. So it can be compared with the role of the hemoglobin in the human body. And concerning more in deep the presence of copper in the human body, we can consider it as a trace element. Copper is present usually in around 150 milligrams in an adult human body. It is present in different percentage in bones, in muscles, in the liver, brain, or also in blood. Also in humans, copper is a cofactor involved in enzyme activity that regulates cells growth, respiration mechanisms, and hemostasis. So copper is considered a very important element, and the deficiency of this element in human is linked to some diseases like anemia or a rise in cholesterol, damage the muscles, and when it comes to heart muscles, it can also be correlated to blocked artery and hypertension. And some studies also reveal that there is an increase in bone fractures in the case of copper deficiency, which is linked to the problem of osteoporosis. As I mentioned before, copper is involved in enzyme activity. So on the right, I reported two enzymes that works by bonding copper. The first enzyme is the superoxide dismutase, which has the role to mitigate oxidative damage outside the mitochondria. And this enzyme is able to bind the copper to ion and zinc together in order to then bind to reactive oxygen species like the oxygen species reported on the right, and to neutralize these reactive species by transforming them into peroxide water, peroxide, and then ultimately into water. The cytochrome C oxidase on the bottom, on the other hand, it has a very important role for giving energy to cells. In fact, cytochrome C oxidase is an enzyme involved in the production of adenosine triphosphate, named ATP, which is the main source of energy for cells. But how do we can we have copper in our body? How can we introduce copper in our body? Well, the main source is of course food. Here I listed some food that are rich in copper. For example, we can find this element in nuts like cashews and peanuts that contain respectively 0.6 milligram and 0.4 milligram of copper. But we can find copper also in dark chocolate that contains 0.4 milligrams for 22 grams of chocolate, and also in some fish like salmon. But the richest food in the richest percentage of copper can be found in the liver. Liver contains 12.6 milligrams out of 77 grams of products. And for all the people that are following a more vegetarian or vegan-oriented diet, also potatoes and kales can represent a source of copper. So after we eat these nutrients, the main organ involved in copper absorption is the gut. Inside the gut, after the copper is absorbed, it can be complexed to albumin and enter inside cells, where some specific proteins called copper chaperones bring copper to mitochondria and the Golgi apparatus. And after the blood, the copper is complexed and brought by blood, it reaches the liver, which is the main responsible of copper distribution. In fact, from the liver, copper is redistributed into muscles, or 23% of copper is reported inside muscles, brain, kidneys, and also other parts of our body. The liver is also the responsible of copper excretion through the biliary route, and it is estimated that around 0.4 milligrams of copper per day are pulled off our body through the biliary route. Now let's dwell more into the use of copper in microbiology and in biomedical devices. So copper is actually an element which is already present in the life of bacteria. Around 72% of bacteria appear to utilize this element for enzyme activity. But actually this element can also be toxic when it is present in high concentrations. So actually bacteria developed a way to cope with an excessive amount of copper, and they also possess some metabolisms and abilities to pull the copper out pull the copper away, and so as it's not too toxic and it cannot be harmful. But the response that bacteria have to copper is different depending on the composition of the cell wall of each bacteria type. In fact, bacteria divide into gram-positive bacteria and gram-negative bacteria. The gram-positive bacteria have a cell wall that is made by the following layers. So starting from the bottom, there is an inner membrane, and followed by the periplasm. And after that, there is a thick peptidoglycan wall where teichoic acids and lipoteichoic acids are inserted. A typical example of a gram-positive bacteria is Escherichia coli with its elongated morphology. On the other side, the gram-negative bacteria have a cell wall that from the bottom has an inner membrane, then a peptidoglycan layer, but they also possess an outer membrane where lipopolysaccharides are present, and where we can find also some membrane proteins like surface proteins and porins. And a typical example of a gram-negative bacteria is the Staphylococcus aureus. So, as I mentioned before, these two types of bacteria develop different mechanisms for coping with copper. For example, gram-positive bacteria have an intracellular copper sequestration that consists in having specific proteins that bind copper when it is already inside the cells, and they prevent the excess of copper to create damage to the cellular components. The second mechanisms they developed is copper transporting ATPases that are able to take to bind the copper and to pull it outside of the cell, while the extracellular sequestration is a mechanism that is able to recognize the presence of copper outside of the cell and avoid the copper to enter the cells. The mechanisms of the gram-negative bacteria are periplasm mismanagement, efflux pumps, and siderophore-mediated sequestration. The periplasmis management, again, is a mechanism that binds the copper when it's inside the cells and pulls the element outside, as well as the efflux pumps. While the siderophore-mediated sequestration it involves the reduction of the copper 2 into copper 1, avoids the reduction from copper 2 to copper 1, which is considered more toxic. If we want copper to become active against bacteria, because we want to fight the microbial resistance, we have to find some ways of optimizing experimental conditions in order to make copper too toxic for bacteria in order to kill them. And one way that is under study is the usage of copper nanoparticles. These copper nanoparticles have the ability of killing bacteria by two principal mechanisms. The first one is the cell wall disruption, and the second one is ROS generation. And in fact, if we have a further look at the image on the right, when copper nanoparticles enter bacteria, they are able to generate a lot of reactive oxygen species that are able to damage the DNA and consequently also cause protein damage and mitochondrial dysfunction. And also the shape of copper nanoparticles is able to disrupt the cell wall. So I reported on the bottom an image of scanning electron microscopy that shows the cavities in bacteria cells walls of Escherichia coli treated with the copper nanoparticles. Despite copper looks a very promising route for fighting microbial resistance, actually it has also an environmental impact that we cannot avoid. So copper is known to be toxic to aquatic life, and it's also known to be able to contaminate the soils. So this can represent a very big problem for crops and for vegetables that become the food we eat. And in fact, the excess of copper is also linked to human health risks. The water quality degradation and wildlife are other two problems that are caused by the contamination of copper. And as I mentioned, the resistance in the development in the antimicrobial resistance is one of the biggest challenges that we have to fight. And in fact, in case of an excess of copper, bacteria can develop even more strong reaction to defend themselves from copper. So we have to be careful in the usage of this element in order to make it an advantage for us and not a disadvantage. So thank you very much for your attention, and that's all.